Yeah, good morning and a warm welcome from Avanto and Beckman for today's webinar. So thanks for joining us today. Our topic for today is optimizing protein and virus purification using ultracentrifugation. So you choose the right webinar, that's for sure. But before we start, and I hand over to Abraham, just share a couple of housekeeping notes. If you have any questions, don't hesitate to raise them using the question area. We are happy to pick them up and answer that in the best way as we could. While we have at the very end of this webinar, we have a question and answer section. So we have foreseen a couple of minutes for your questions. But if you have any related to some particular slides from Abraham, don't hesitate to raise them. We take care about that. You don't have to write something down, or keep in mind, as we share the recording link right after the webinar with all materials that we are sharing during that webinar. So don't worry about that. That will be shared in the follow-up email from avantrosciences.com. Yeah, having said that, I will hand over to Abraham, which is your host today. A welcome to you, Abraham, and thanks for joining us and giving us this valuable insights today about this optimizing of the protein and virus purification. So thanks, and the stage is yours.
Thank you very much. I'm Abe Wilson. I manage the technical product manager team at Beckman Coulter on centrifuges. And today, the focus of our conversation will be on ultracentrifugation and how we can use it. I'm very, very excited to talk about this. There's some legal notices I'm told I have to share, but let's jump right into the agenda. Right, I'm going to start off with a little bit of a background. I think we're only going to spend less than 5 minutes on the background here, kind of the introduction, since I assume there's a more familiarity on this already. But then we'll go into what are the levers that we can adjust to optimize density gradient ultracentrifugation. We'll talk about optimizing specifically isopycnic, these density resolved gradients that are commonly used for virus. Then we'll talk about optimizing rate zonal gradients. And then finally, I have a case study kind of tying this all together, comparing ultracentrifugation to anion exchange. So let's jump right into it. We'll talk about the what, why, when, and how. First, what is density gradient, right? This is a purification process that separates particles based on physical properties. And those physical properties can either be the size and mass, we call that S value, or the density. You obviously do this in the presence of centrifugal forces. And the separated species form what we like to call bands, right? And this is all conducted in a column, kind of liquid with varying density and or viscosity. And there's a distinction here that I will be mentioning more later on, right? But the gradient viscosity will affect the particle migration rate, so how fast the particles move through the tube. That has certain ramifications. And the density will affect where they will end up over time. Okay, so we will be talking about both. Okay, why is this so commonly used? Right? Density gradient purification is chosen for difficult separations that require both high purity and yield. And you know, right now I'm actually at a trade show for cell and gene therapy. Right there on the far left, right, viruses. This is where this is most commonly used. I say right now, this is a very commonly used right there, but this has been used for a very long time, 80-ish years almost, for all kinds of other products. Right, for proteins, plasmids, etc. Not going to go into all the details here, but we will be talking today predominantly about viruses and also about nanoparticles and to some degree proteins as well. Okay, where in the process do we do this? When do we do this? This is typically used for downstream purification, right after an affinity chromatography step or after lysis directly. Right, the process to density gradient ultracentrifugation, I think, it boils down to just 3 steps. Right, it's not super complicated. I like to think first you have to form the gradient. You're choosing the gradient material. You're choosing the gradient method. That's kind of the how of the process. That's on our next slide. And then you choose the rotor. You choose the tube sealing format. You choose the tube material. You choose your rotor, and then you choose how you retrieve your sample. Right. Everything really boils down to these three. We're going to talk about optimizing each one of these steps. But let's talk about that method a little bit more. So how does this process work? Well, there are different gradient types. And I think to start us off, it's helpful to ground us on actually not a density gradient, but pelleting, right? The most commonly used centrifuge method is pelleting. Materials separate by S value, which is their size and their mass, as they sediment to the bottom of the tube. Right? There's no density gradient that's used. I have a picture of a blood cell there, right? This is fast and simple. This is commonly used for cell harvest or for separating cells. Started out, this is how centrifuge started, right? It was separating milk and kind of the heavier milk solids from the whey. But let's get into the density gradients, right? Ray zonal is similar to pelleting in the sense that the materials are also separating by their size and mass, but they're doing so in the presence of a density gradient. And this is a continuous gradient. Commonly it's sucrose, but there's other media that we'll get into later. This can offer a very high resolution, rate-based separation. And so proteins, right, and protein aggregates where they have differing masses of just even small degrees, that's where you get a very good separation. This is a foot race. Who can get to the bottom of the tube first? But before it pellets, you stop the run, so that's why you have those discrete bands. That's ray zonal. Let's move on to isopycnic. Isopycnic materials are separated by density, right? And they're doing so in the self-forming gradient. Commonly, they're using a salt of some kind, like cesium chloride or iodixonol, which is not a salt, but so you're doing this in where the bands will then end up where the density gradient media is exactly equal to the density of your sample. That's where the sample will migrate to, and this is the highest resolution density gradient separation done by density. There's kind of an in-between method here too. Some scientists call this equilibrium zonal. It is still an isopycnic method because the materials are still separated by buoyant density. But instead of in a continuous gradient, you're now doing this in a preformed density gradient, and you don't even allow the materials to get to equilibrium. So in that sense, it's similar to a rate zonal experiment. The sample goes through these kind of discrete chunks of density. And so at the lower density areas at the top, you might have all your junk, your lysate, etc., and your sample might migrate down to a lower area, to a lower layer. This has the distinct advantage of being fast. And so you have a one-step purification and concentrations process by density. That's our introduction. Let's talk about the levers to adjust, right? So as we think about optimizing density gradient runs, there's really just four things that we can optimize, right? There's the density gradient itself, the media. That's what I'm going to call the density forming material. You have the rotor, you have your tube, and you have your centrifuge. And we'll talk about how we can optimize these and then what are going to be some of the ways we do this for specific samples. So first, you have to choose a density gradient media based on what type of density gradient best suits your sample. Right? This is mostly meant for you to screenshot, right? Because on the far left-hand side, I have a list of different density gradient medias, and each one of these medium, these are commonly used, I think, but then this is just something that has been based on my and my other scientists' experience on reading a lot of articles and what we commonly see in the field. Those useful max density numbers there are guidelines, I'd say. It can be higher than that if you are doing certain things, but this is generally what we're seeing. And then you can see based on what kind of sample you have, you're going to be choosing a specific kind of density gradient media. Okay, let's talk about the rotor a little bit now. The rotors have distinct advantages based on what you're trying to do. And so it's important to understand what each rotor type offers you. Right? There's the swinging bucket rotors. For these, the tubes that the sample is in actually swings out and it goes perpendicular to the axis of rotation. It has the 90-degree angle. Right? These are best for rate zonal and pelleting. runs, right? They have the advantage of being able to maximize the pellet stability because you can even use a conical tube and everything can be concentrated into just this small area at the bottom of the tube. Path length is how far the materials have to travel in the centrifuge, right? And it is long, right? Because they travel the whole length of the tube. Okay. The next rotor type is fixed angle, right? And this is, kind of halfway between a swinging bucket and a vertical, right? It is extremely versatile. It is still commonly used for pelleting, and it has the advantage of being a little bit faster because now that path length is shorter. Now that has some consequences, right? You may have, if you're concerned about shear effects, you might be using a swinging bucket rotor where the sample will not be hitting the side of the tube as much. So there are some consequences to that, but it is faster, and this is generally a very versatile rotor. Kind of moving in this direction, there's the near vertical, right? Now you're getting to extremely close to vertical so that the run times are getting very, very short. It's not great for pellets because you don't get as good of an area to collect the pellet, but you can clear out contaminants. So this is very useful for purifying, for example, crude plasmids. And then finally, you have your vertical rotor, where this is used for isopycnic density gradients, basically exclusively, right? And you're separating by density, and you can have very fast run times because the path length is very short. Okay, so you kind of go from pelleting and rate zonal there on the left to isopycnic separations there on the right. Okay, next let's talk about our tubes, right? Again, I'm not going to actually go through everything on this slide, but I hope that this is screenshotable for you, right? There's different tube types that are important based on your chemical compatibility, right? And so I'm not even going to go through them there, but you got polypropylene, ultraclear, and polycarbonate. At the top there, I've listed the different sealing types. Okay, so quick seal offers an excellent kind of permanent heat seal that's good for secondary biocontainment. So if you're dealing with hazardous material, right, a lot of our viral customers are using quick seal. With that kind of permanent seal also comes having to make that permanent seal. And so there's a little bit of time associated to that. If you're sensitive to that, then we recommend the OptiSeal, which is an easy finger touch seal. The wider neck is also very easy for the extraction portion later. And then finally, we have open top tubes that are easiest to fractionate from the top, right? We also offer some sterile and endotoxin-free tubes to help out with the workflow. Based on what your sample dictates, right, you're going to be choosing one of these different tube types, okay? Finally, we come to the centrifuge. And the centrifuge is a little sneaky, right? There are certain things here that we can do that can help with your run. There's obviously, the speed, the temperature, the time, the capacity, right? And so here's the speed, right? Certain speeds are going to be required for the gradient to form, but you can also have multi-speeds in the experiment. We're going to talk more about that later. Temperature, colder temperatures may require longer run times. There's time, right? Spinning too long in the case of a rate zonal or equilibrium zonal. You might even get a pellet, might be bad for you. It may just not be necessary. Or too short, right? You may not be fully reaching equilibrium, so you have to be cognizant of that. Then finally, capacity, right? There's different capacity centrifuges, obviously, and so we have to be cognizant of this. So I am going to go through these considerations here, right? You can change speeds mid-run. I mentioned that. You could also change your sample concentration, right? In general, I think it's great to minimize the amount of centrifugation we have to do here, and so the degree to which we can concentrate sample beforehand using something like TFF can be very helpful. And then finally, gradients can also be formed to some extent outside of the centrifuge, and that can really cut down on the centrifuge runtime, and we're going to talk about that. Okay. At this point, we wanted to launch a poll to get a better understanding of what samples we're primarily working with.
Yes, Abraham, coming up. Perfect. So just share what you are processing in your day-to-day business. That would be a great information for us. So please vote now. Okay, results coming in, Abraham. Just waiting a couple of seconds more. Everybody can make his choice. Okay, looks like a clear picture, more or less, Abraham. So 64% are using protein samples, 36% are going for the virus, and 27% do some other samples or processing other samples. So 60% protein, 40% roughly virus.
This is terrific live feedback. So we might go through this virus portion a little faster then, so we can dwell a little bit more. Oh, sorry, my screen here went down. So here we are. This is terrific. So yeah, here we are on the viruses, and then we'll get to the rate zonal next, which will be more applicable to the 64% of you on proteins. Thank you for humoring us there with the poll. We're going to review the levers, I'm sorry, here for the viruses. What are we going to be changing? And then I'll talk about two of the levers in particular, the rotor and the gradient, and then we'll get to the summary here at the end. And I've got case studies kind of scattered throughout here as well. So for viral purification, we really want to consider four things. First, a lower solution viscosity, right? Reducing solution viscosity here will improve performance. Commonly, we're using benzonase, right, to digest nucleic acids. Second, I mentioned this just a few slides back, right? We want to be concentrated on our sample. We want to be reducing the total amount of centrifugation required. Something like TFF here can be very helpful. I think these are more commonly understood. Now, though, we get to what I think a lot of people could be doing to help out their run times, right, is we can be facilitating gradient formation. And when dealing with virus, right, you can really shorten your path length by reaching you can reach equilibrium faster by shortening your path length. You can also use, to some extent, a preformed gradient, right? So we'll talk about that actually on the next slide. And then there's speed, right? We can change speeds in the middle of the run, and this is very important to be able to form that gradient as quickly as possible. Okay, let's talk about this path length, right? Why does this matter? The separation of the empty and full particles in cesium chloride is faster using a vertical rotor. So I've got it just as an example here, the swinging bucket at the top and the vertical rotor at the bottom, right? We're just going to assume it's all homogeneous cesium chloride. And while spinning in the swinging bucket rotor, you can see that the particles have a longer distance to travel. And as a result, the gradient forms more slowly. By contrast, the vertical rotor, that gradient is going to be pretty shallow, right? There's just not much distance for the cesium for the cesium chloride particles to go. There's not they don't go very far, and as a result, you can greatly decrease the amount of run time. Okay. What will happen after it's out of the centrifuge, right? The gradient will reorient in a vertical rotor. I'll actually show you that a little bit on the next slide. And so they end up forming a gradient the same way at rest. As you can see, it starts at white at the top, goes to black at the bottom. That's our fancy visual for gradient here. But there's something else that's very important about these vertical rotors that is relevant to virus separations, and that is that you can also improve your resolution. Okay. By virtue of the fact that you're forming a very long path length, wide density gradient in a swinging bucket rotor, you can see the minimum density gradient is 1.05 grams per ml to about 1.45 swinging bucket. By contrast, in a vertical rotor, it's going to be much tighter. You basically are putting a magnifying glass just on the region of interest for you, right? And these numbers will obviously change based on how you're diluting your density gradient material. This is just for illustrative purposes. This is commonly what we're doing when we're separating virus. When it is in the rotor, right, in a vertical rotor, it is forming vertically, but then when the bands come to rest, the amount of volume in between the bands remains the same, but because the tube is longer than it is wider, there's going to be an expansion of the distance between the bands, right? This happens because of the reorientation of the gradient. And so just as an example, right, the 39 mil tubes that are commonly used for virus purification, they are 3.5 times longer than they are wide. And as a result, the distance of the bands is about 3.5 times greater. And there's an expansion of about 3.5 times in a vertical rotor when they're in the centrifuge versus out of the centrifuge. And this is just a much better resolution than you can get with a swinging bucket rotor. And so these are the notes there. This is just repeating what I just said there, right? The shorter path length leads to a shallower density gradient. That's the magnifying effect. The shallow density gradient has a smaller range, meaning there's little change in density from one side to the other. And that means that you're able to really hone in on your sample of interest, and then also the density gradient reorients, allowing you to see that better. So what does this look like kind of in an experiment? I'm sorry, before I get to the experiment, right, I just wanted to also point out that there's the VTI 50.1, which has almost half a liter of capacity, right? So there's a very high, you can do pretty high throughput with this as well. So what does this look like in an experiment, right? A vertical rotor we were able to actually see a band where other rotors weren't able to. We worked with a partner, CBM, Center for Breakthrough Medicines. They're a CDMO in America, but we worked with them on separating adenovirus, and we ran these in different rotors, all trying to reach equilibrium. As you can see, these are relatively long run times, right? And this is the end product. Right? Down there on the bottom left is using the existing protocol that they had with the 50.2, where we were able to see one band. But then when we used the VTI 50.1, we actually saw a third band there. And so this is just illustrating that improved resolution that you're able to get with a vertical rotor. Now, those bands do exist in the other rotors, but you're not able to distinguish between them. They look like one band because the density is so similar there. So with the shallower gradient in the vertical rotor, you're able to better separate. All right, let's now talk about the gradient. Because density gradients form before the sample fully migrates, by layering sample on top of the gradient and then running at a higher initial speed, sample can be purified significantly faster. Now, there's a lot to unpack in that sentence, and so I hope to do so with some graphical kind of interpretation here. The top there, I've got sample homogeneously scattered throughout the tube with the gradient forming material. And in these graphs, right, I'm showing density on the y-axis and then just the radius of the tube in the centrifuge on the x-axis there. And on the bottom, right, we're going to assume that you put all of your density gradient forming material at the bottom. If that's cesium chloride or iodixonol, I don't care, right? It's all there at the bottom, and then you put your sample just at the top of the tube, right? And so you can see the sample is less dense than the concentrated gradient material, and so that's why you see that separation there at the bottom. If we just look at the top here, though, for just a moment, right? Inside the centrifuge, that gradient will form, and it will form slowly, right? And as it forms, the sample is going to move from both sides of the tube. They'll start to form distinct peaks, and at the end of the run, you'll get a profile, and you'll get your empties, you'll get your partials, you'll get your fulls. It might look something like this free-form sketch that I did here in PowerPoint, right? But if we go down to the bottom now, if you look at this instead, where all your gradient is, just at the bottom of the tube, and then it forms a gradient faster because you've already started forming that gradient just by having one step. You could have multiple steps and it will form the gradient even faster. It forms that gradient faster, and it forms it steeper if you're doing this at a higher speed as well. And that higher speed will enable you to form this gradient very quickly. But that's not what you want, right? The sample peaks are now closer together because you formed a steeper gradient. So if you then decrease your speed, you can end up with the exact same profile, but you can just get there faster. And again, I've got a little case study of something we did in our lab at Beckman Coulter, right? We formed a linear density gradient in 80 minutes using a vertical rotor. Doesn't matter really what sample we used AAV here, and then we have the cesium chloride at the bottom. In this instance, we used the VTI 90 vertical rotor. And our known protocol that we were comparing against was a 16-hour run, right? But we were trying to get this time down, and we used actually about 6 different test times that we were working with. I'm only showing 2 here where we ran it at 90K RPM, so at max speed for either a half an hour or 50 minutes. And then we ran it at a slower speed at 60K to try to get to the exact same density gradient profile. And then we fractionated the whole tube, collected these fractions, and then did radial interference to measure the gradient. And this is what we get, right? That left-hand chart isn't as instructive, I think, as the right-hand chart, right? So we have that known protocol, the 16-hour protocol. That's at the 0 line. And then we compared all of our kind of experimental protocols against the known protocol. And what we wanted to see was no change, right? We wanted to get the exact same density gradient because then our sample would be in the exact same spot, but we could just get there much faster. And what we found was that running it at 50 minutes at 90K RPM and then 30 minutes at 60K RPM, that's the red dots there, got the exact same profile as that 16-hour run. And you could see the difference there. It was close enough for us to feel very comfortable. By contrast, that 90-minute run where we ran only 30 minutes at max speed at 90K and then 1 hour at 60K, we didn't feel comfortable with that gradient, right? Yes, the difference between it and the known protocol, if you look at the y-axis there, it's not great. It's not very big difference, but we still weren't comfortable. We thought, okay, this is going to actually have impact on where the sample bands are. And so we prefer the red. But the point is that you can achieve the same exact density gradient profile in significantly less time. I think that's terrifically exciting, and I just think that's really cool. So there we are. Use the multi-speed parameters that are available to you in many different centrifuges, such as the Optima XPN. So let's put this all together. We're running AAV experiments, right? We're going to concentrate and reduce viscosity as much as possible in a cesium chloride gradient. We're going to layer the sample above the cesium chloride, use a vertical rotor, use the multi-step experiment. If we're using iodixanol, we're going to be roughly at the 1 to 1.5 ratio for iodixanol. And when you have those step gradients, the vertical rotor can offer marginally shorter runtime, but you still have some degree of benefit there. Okay. Now let's talk about rate zonal separations. Right? And I'm sorry, my example here is going to be for nanoparticles, but the same principles are going to apply. And I know we got most people here who are doing this with proteins. So this is going to be simple, right? We're going to talk same kind of thing here, levers, right? And then we'll talk about the concentration, sample concentration, and then the path. So let's look at these factors. Again, higher viscosity slows sedimentation. In this situation, that can actually be a good thing, right? Because you don't want in a rate zonal separation, you don't actually want your materials to pellet. And so if they have a high S value and they are pelleting very quickly, right, you can choose to have a higher viscosity solution to help you with that. Sometimes it does slow your runs down, so it's not necessarily always a good thing. Again, in that same vein, temperature has that same impact, right? The colder the temperature, the longer the run. And now we get to the 2 points that I'm going to spend more time on, right? Sample volume and concentration. Larger sample volume can lead to more diffuse bands. I'm going to talk about that on the next slide. And then path length. This is the opposite of everything I just said for the viruses, right? The longer the distance now, the better the resolution, and we'll talk about why that is. Let's jump right into the sample volume and concentration, right? The more concentrated the sample, the less potential separation distance, and the more viscous the media, right? So with a diffuse band, what we're basically doing is we're starting starting sample out at many different spots, right? This reminds me, I ran one marathon. I'm sorry, I'm going to use a metaphor here, right? But this is the functional equivalent of somebody starting, a mile apart in the same marathon race. They might be the same speed, but they're going to finish a mile apart, right? And so if you have a large sample that you're starting with, you're going to end up with more diffuse bands. Right? By contrast, if you're able to have a small, highly concentrated band, then you're going to have smaller bandwidth, and that will be a little bit easier for you to separate and diffraction. If you want to incorporate more sample, then we should be using wider tubes, right? That will allow for more sample while still minimizing the band height. And an example of that, I'll show you this case study here with nanoparticles. This is not something that we did in our lab at all, but I'll just cite this here, right? This is separation of nanoparticle variants. This, I believe, came out of the University of Constance. And they used rate zonal separations here to separate 12 different nanoparticles that vary by their sedimentation coefficient. And here they pre-layered a sucrose gradient, right? That's saving the time. And so then they were able to have just a 15-minute run, and then they're overlaying just 2 mils. Like I said, it's not a lot, but they're doing that because you're getting much more concentrated bands, and then they're using the widest possible tube and rotor, that's the SW32, so they're able to put in more sample by using the wider tubes, and then they get the 12 distinct bands, as you can see in their image. Okay, the same principles here will all apply to proteins, right? Proteins and protein aggregates is going to be the same thing. Okay, let's talk about path length here. Right now, the longer the path length of the tube and the rotor, the better the achieved separation. This is the same principle as old school sequencing gels, right? The longer the gel, the better the separation. It's just because you're allowing more time. So if I go back to that marathon metaphor, right? I am not a fast runner. And so I've done one marathon, but no, I'm not going to say that I was very good at it. The longer that run went on for, the farther the distance was between me and the fast marathoners, right? And so with that same principle, right, the longer the distance that we have to go for the samples have to go, the greater the distance will be between the bands. Okay. So there is a trade-off though, right? There's also going to be the longer runtime. So you may not need to get that great of a separation if your fractionation or your sample extraction is good enough for you already. Then that's something you have to be aware of. If you want to have a faster runtime, you could use the fixed ankle rotor, right? I mentioned earlier, this is the most versatile rotor, right? And so you can get a faster runtime by having a shorter path length, but your bands will be closer together. That's not always a bad thing, so long as you're able to effectively separate the sample. That's what we really need. Okay, we're now going to put this all together with a density gradient example, comparing this to anion exchange, right? There's trade-offs here. In this example, we used adenovirus, and I'll walk you through our case study here just a little bit, and then we'll have the summary. We'll be done. We'll have time for some questions if we need them. The objective of this case study was to compare anion exchange to density gradient ultracentrifugation to see what's the impact on recovery, purity, functionality, and other critical quality attributes, right? I'm not going to get into the upstream process. We did start with 2 liters, but once we got to the lysate clarification, we separated 50%. We sent to the chromatography, and 50% we sent to the centrifuge. For the ultracentrifuge, we purified these in the open top tubes, and we layered the heavier, the more dense material at the bottom, 3 mils of that, and then a little bit less dense material above that, and then we put sample on top of that. With these tubes, it's important to leave 1 to 2 millimeters at the top, so you're not filling this all the way. That's just specific to these tubes. What that meant is that amounted to about 5 mils of adenovirus for us here. We used an SW41 Ti, which, if you've been paying attention, is not necessarily the best rotor. Here we were limited by what we had with our partner. This was we partnered with Sirion Biotech, so we were very grateful to them. They have a lot of expertise here with adenovirus. But you can see we were practicing that density layering to shorten our runtime. Here we use the swinging bucket rotor, and we spun it at 1.5 hours. And then we just syringe extracted the 1 mil-ish of adeno, took it down for polishing, and then analysis afterwards. Okay. I will point out too, the adenovirus that we were working with, this will depend a little bit on your gene of interest, but our density was 1.35, which my understanding is quite common. Okay. This is the AEX workflow, right? We had a flow rate of 4 mils per minute, ran it for an hour, and then we pooled the fractions. Okay. Here's the results. What we wanted to do was we were looking at the yield, obviously. On that far left there, we were looking at infectious units per mil, and then on the right there, we're looking at the viral genomes per mil. The y-axis is a logarithmic scale. We did that to be able to compare both of these. Density gradient ultracentrifugation yields were significantly more, right? About on the order of 200 times more. And we also wanted to convert those values to look at the total particle counts, right? And it's even greater when you look at the total particle counts, right? We summarized kind of the output here in this specific table, right? In terms of final product, we were able to get 4.5 mils with density gradient with a much higher infectious particles per total particle count than we were with the chromatography method. Now, chromatography also comes with its advantages, right? The ability to scale up is much better, but we were able to get just a much greater yield and recovery with the density gradient method, right? Oh, I'm sorry, I'm not sure why it just ended there, but we have about a 582-fold increase in the number of infectious particles recovered, and we had a higher ratio of infectious particles to total particles, right? To talk a little bit more about infectivity, right? We transduced the cells with purified adeno at different multiplicities of infection, right? And after incubation of 2 days, we isolated the nucleic acids using PCR for quantification, right? Unfortunately, the low concentration of the AEX precluded some of the 200 multiplicity of infection there. So we don't have any data from there, but even from the data we do have, right, you can see density gradient was able to achieve about 28% higher functionality. Then we also did some testing for endotoxin and other contaminants, right? No bacteria, no mycoplasmas in either. Interestingly, we did see some degree of endotoxin with AEX and none detected in the centrifuge. But then we did see some residual host cell protein in the centrifuge. When you account for the concentration, though, of the centrifuge, this would go to the no detection level. If you were doing that for the anion exchange chromatography as well, right? It was well below the limit of detection when diluted. Which remember, right, that the centrifuge has these extreme concentration effects. So there we go. There were some things that I think we did very well in this experiment, right, in terms of optimizing for runtime. We showed the different layers of gradient. We already had a pretty short runtime. There are some things that we could have done better as well, right? From the centrifuge perspective, we could have been using a rotor that was more suited for this purpose, right? But I hope this shows that the rotors are to some degree versatile in terms of what they can do. All right, I've just got 2 more slides here, just putting this back together, right? Remember, for the viral purification, we're seeking for a lower solution viscosity, a higher concentration, a shorter path length, and a variable speed run. And as we do these things, we can improve our run times and hopefully make this process less painful for us. And when conducting rate zonal experiments, right, again, we have to consider solution viscosity. It may go either way here. We have to consider the temperature again, may go either way here. But then we really need to be thinking about our sample volume, how concentrated this is, and our path length, right? The longer the distance, the better the resolution. Thank you, everyone, very much. I do want to call out some of my help here. The scientific team at Beckman Coulter is excellent. And our partners, Sirion Biotech, right? And I should also have called out Center for Breakthrough Medicines here. I left my email there, awilson06@beckman.com. Feel free to reach out with any questions. We would be happy to address them via email if ever we need after this. But I think also right now we've got time if there's any questions that we need. Thank you so much.
Yeah, thanks, Abraham. So Frank speaking. So let's see what will happen in the question area. If there is any. So Abraham
easy
seems.
on me. I like this.
Yeah, no questions there. So that means vice versa. You have done a great job, Abraham, I guess, right? So, but if there is any question left in your day-to-day business, as Abraham told you, please reach out to us or Abraham. If you want to contact Avantor, then choose the address webinar@avantorsciences.com. We are happy to forward to our experts and to the Beckman experts all your questions you might have after the webinar. If you have interested colleagues as well, which we could imagine that there might be some out there, so please don't hesitate to forward a link you will receive in the follow-up email, which you will receive very, very soon, together with a short survey where we need your input, where we like to have your input on the current session. How was your experience with that? So don't hesitate to share your opinion there. And if there is no other question appearing, just waiting for the Beckman colleagues, if they have received any. So that doesn't seem to be the case as well. So having said that, thanks again, all participants, for joining us today. You will receive, as I said before, the follow-up email of that conference. Thanks, Abraham, for taking the time and sharing all this valuable and valid information with us and putting us through this journey. So thanks for taking the time again. And with having said that, I say goodbye to the audience. Goodbye, Beckman. Goodbye.
Thank you.